All right, welcome in, future nurses. In today's explainer, we are going to take a walk through the absolute marvel that is the mammalian cardiovascular system. As you get ready to step out onto the clinical ward, really grasping the biological mechanics of the heart and blood vessels, well, it isn't just some academic exercise. It's literally the foundation of everything you'll do in patient care. So today, we're going to treat the cardiovascular system like a biological engine. We'll look at how it hums along when it's totally healthy and exactly how we measure its performance when you're standing right there at the bedside.
Okay, let's run through our agenda for today's explainer. 1. The mammalian double circuit. 2. Heart rate and cardiac output. 3. Blood pressure and vasodilation. 4. Blood composition and clotting. And finally, 5. Understanding cardiovascular disease. Let's jump right into Section 1, the mammalian double circuit.
So mammals, unlike fish, which just have a single circulation loop, use a highly efficient double circulation system. It's actually brilliant. The right side of the heart is dedicated entirely to pumping oxygen-poor blood straight to the lungs. We call that the pulmonary circuit. At the exact same time, the left side of the heart is pumping all that fresh, oxygen-rich blood out to the rest of the body. That's your systemic circuit. So the big takeaway here is that our four-chambered heart perfectly separates these two loops. It's basically two coordinated pumps fused into a single organ, giving us a really vigorous, high-pressure flow of oxygenated blood to feed our energy-hungry brains and muscles.
Now listen to this statistic because it's genuinely mind-boggling. Inside every single adult patient you treat, there is enough total length of arteries, veins, and capillaries to wrap around the earth twice. Twice! It perfectly illustrates the sheer scale of the microscopic plumbing network that we are keeping alive with every single heartbeat. Delivering oxygen through a maze that massive takes an incredibly reliable engine.
Which brings us nicely to Section 2, heart rate and cardiac output.
For a resting adult, one complete sequence of pumping and filling is fast. We're talking just 0.8 seconds. It starts with diastole, a relaxation phase. Blood returning from the large veins just sort of effortlessly flows right into the atria and ventricles. Then, at 0.4 seconds, we hit atrial systole, which forces any remaining blood down into the ventricles. Finally, at 0.5 seconds, ventricular systole kicks in, powerfully slamming that blood out into the major arteries. This whole flawless dance happens in fractions of a second, totally governed by the heart's own internal pacemaker, the sinoatrial node.
So before we get into the physical clinical measurements you'll take on the ward, let's ask ourselves a core physiological question. What actually dictates the heart's overall workload? How do we quantify how well this biological engine is performing? The answer, and please lock this definition into your brains for your nursing practice, is cardiac output. Cardiac output is the total volume of blood that each ventricle pumps per minute. How do you find it? You just calculate the heart rate, so beats per minute, and multiply it by the stroke volume, which is just the amount of blood pumped out in one single contraction. This is absolutely crucial because it tells you exactly how effectively your patient's heart is delivering vital oxygen to their tissues. To put that into a relatable perspective, 5 liters per minute is the resting cardiac output of your average human. Now, the average human body only holds about 5 liters of blood in total, which means essentially the entire volume of blood in your patient's body passes through their heart every single minute. And if they start doing heavy exercise, that output can shoot up by 5-fold. It's just wild. Let's move on to Section 3, blood pressure and vasodilation. Okay, when you take a patient's vitals, you're always looking at 2 distinct numbers. Let's clearly define them. The first, that top higher number, is the systolic pressure. This is the arterial blood pressure generated specifically when the heart contracts during ventricular systole. It is literally the absolute peak force pushing outward against your patient's arterial walls during a heartbeat. In fact, the arteries actually stretch out to a wider diameter just to accommodate that massive surge of pressure. But while we have that intense peak systolic pressure, it's paired with the diastolic pressure. During diastole, the heart relaxes, but those elastic arterial walls, they aggressively snap back. That recoil is vital because it maintains a continuous, lower pressure between beats. It ensures blood keeps flowing steadily into the tiny arterioles and capillaries, even when the heart is taking its brief resting and refilling phase. Now, the body carefully regulates all these pressures using smooth muscles wrapped around the arterioles, which introduces a massive concept for us, vasodilation.
Very simply, vasodilation is an increase in blood vessel diameter caused by the relaxation of those smooth muscles in the vessel walls. The crucial takeaway here is that when vasodilation happens, usually triggered by chemicals like nitric oxide, it lowers the blood pressure upstream, which allows an increased rush of oxygen-rich blood to specific tissues, like when your muscles need it during a workout. And of course, the opposite is vasoconstriction, which narrows the vessels and drives blood pressure up.
We also can't forget about gravity. When your patient stands up, their brain is physically way higher than their heart, right? So blood literally has to fight gravity to get up there. If their blood pressure drops too low to make that uphill climb, they faint. And here's the thing, fainting isn't just some quirky clinical symptom. It's a drastic homeostatic rescue mission. It collapses the body to ground level to instantly put the brain on the exact same gravitational plane as the heart, rapidly equalizing the pressure and restoring that critical oxygen flow.
So how do we track this? At the bedside, you're going to use a sphygmomanometer. You'll inflate the cuff until the pressure actually exceeds the artery's pressure, closing it off completely. Then you slowly deflate it. The exact moment the blood violently pulses back through and you hear that sound in your stethoscope, boom, that's your systolic reading. You keep deflating until the blood flows totally freely and silently again, and that silence marks your diastolic pressure.
All right, let's step into section 4, blood composition and clotting. Whenever you draw blood for labs, you're really extracting a highly specialized liquid connective tissue. Roughly 55% of that volume is a fluid matrix we call plasma, and the remaining 45% is cellular elements. Now, the plasma is mostly water packed with dissolved ions and proteins. It's the ultimate transit system, handling osmotic balance, buffering pH, and carrying everything from circulating hormones to apolipoproteins that escort lipids around the body. And then we have the cells. Just think about the sheer dominance of erythrocytes, our red blood cells. We're talking 5 to 6 million of them packed into a single microliter. Their one job is oxygen transport. Actually, mature mammalian erythrocytes literally spit out their own nuclei and mitochondria just so they have more physical room to pack in oxygen-carrying hemoglobin. It's amazing. They share that space with leukocytes, which are your immune defense, and platelets, little cell fragments that are the absolute stars of our next topic, clotting. Because if the body's plumbing gets breached, we need a fix fast. Enter the clotting cascade, an incredibly elegant positive feedback loop that quite literally saves patients from bleeding out. First, those platelets rush in and stick to the damaged tissue, forming a quick, temporary plug. Then, a massive enzymatic cascade fires up. An inactive plasma protein called prothrombin gets converted into its active enzyme form, thrombin. Thrombin then acts as the catalyst for the final magic trick. It converts liquid fibrinogen into solid threads of fibrin, weaving a tight structural seal right over the wound. Let's wrap up with section 5, understanding cardiovascular disease. This is where we see the mechanics fail. Take the silent, deadly progression of atherosclerosis. It all kicks off when the smooth inner lining of an artery gets damaged, sparking inflammation. Leukocytes rush to the scene and start gobbling up lipids, especially cholesterol. Over time, this builds up into a fatty plaque, mixing with fibrous tissue. The artery gets stiff and blood flow is severely restricted. But the real nightmare scenario? If that plaque ruptures. Because a rupture triggers the exact same clotting cascade we just talked about, but it happens inside the blood vessel, forming a dangerous blockage known as a thrombus. And the stakes of those blockages could not be higher. If a thrombus forms in the tiny coronary arteries that supply the heart muscle itself, it completely starves the beating heart of oxygen. The cardiac muscle dies. That is a heart attack or myocardial infarction. On the other hand, if that blockage cuts off the arteries supplying the head, it starves the brain, destroying delicate nervous tissue, and that is a stroke. We all know that high levels of LDL cholesterol really fuel the growth of those plaques. But genetics is a huge piece of this puzzle too. When looking at massive sets of genetic data, scientists found that patients with a natural mutation that inactivates the PCSK9 enzyme actually display safely lowered plasma LDL cholesterol levels compared to folks with a normal functional gene. It's a really fascinating glimpse into how our innate genetic blueprints intersect with our overall cardiovascular risk. But here's the thing. While our genetics are vital, our lifestyle choices, getting up and exercising, cutting out trans fats, quitting smoking, those are entirely in our control. So, armed with all this biology, I want to leave you with one final thought. When you step onto that ward, how are you going to use the mechanics from today's explainer to convince your patients that their daily choices are quite literally physically reshaping the inside of their arteries. Something to think about. Thank you so much for joining this explainer, and good luck out there on the clinical floor.